What I want to do right now is do a kind of brief run-through of the basic vocabulary and concepts from biology that we're going to be using over and over again on problems in this class. And then, hopefully, if we have some time, to go through one or more concept tests, just exercising a little bit these basic ideas. And if we need more time, I'll take a little bit more time on Wednesday to go through this. So the obvious challenge here is that, just like any field, CS included, biology has its share of jargon, and we'll really limit this as much as possible, but there are a few basic concepts that we really do need to reuse over and over again. It may sound complicated at first, but I think you'll see as you use these over and over, the basic concepts are quite simple. It's just a matter of getting used to a little bit of new language. So I'm going to go through a quick overview of some basic genetics, some of the data types, which are broadly referred to as genomics, that we'll be working with, and some what you might call basic molecular biology that's needed to understand those data, what they are. So even if you've never been to school, in movies and TV and so on, we're constantly bombarded with the double helix. So since you have been to school, you know that this double helix is basically a molecular chain, two of which can wrap around each other in an opposite form. The basic chain is composed of sugar molecule linked by phosphate, so the sugars are the little red rings, and connected to these sugars are what you might call a four-letter alphabet, consisting of 4 different chemical groups. And the magic of DNA is that these 4 chemical groups actually pair with each other. So the traditional way that we're going to be referring to the 4 types is just by letters A, C, G, and T. A pairs with T, and G pairs with C. So if you know the sequence of one chain, you can immediately read off what the sequence of the other matched chain will be. Of course, when you put together the full blueprint, for example, for making a human being, this data is not just written as a single string. It's written as 23 strings called chromosomes. And geneticists, long before they could read this string, spent lots of time, quote-unquote, mapping the different chromosomes and deciphering what different parts of them did. The total collection of all of the genetic material for an organism is referred to as the genome, the complete DNA sequence of an organism. So, for example, for a small bacterium, that would be about 4 million letters total length, and that would be typically a single circular chromosome. If you go out, for example, to the human genome, now we're talking about approximately 3 billion letters in length, and the functional units scale up quite a bit less. Let's say around 4,000 individual functional units, which I'm going to explain in a moment. We call them genes. In a small bacterium, might have about 4,000 individual genes, whereas the human genome, approximate number, around 25,000, at least of the traditional kind of protein coding gene. Okay, any questions? So, common terminology for referring to these sizes are so-called base pairs for the individual unit of a single letter. And the term base pair refers to the fact that in DNA double helix, they are paired A to T, G to C. And as we scale that up, we have units like kilobases, megabases, gigabases, etc. Questions? Okay, so a few slight complications to the mix. Simple organisms like bacteria commonly keep just a single copy of the genome in each individual. But sexually reproducing organisms like animals and plants keep two copies. And this basically gives you all sorts of benefits, which we'll talk about in a moment. But one crucial point here is that now that you have two copies, they're not necessarily identical, right? And the differences between them, biologists use the term polymorphism to refer to the idea that a single place or gene within the genome can have different variants that differ basically in the letter sequence that we just described. So one A might be now a C in one particular individual chromosome versus the other copy of that chromosome. The term that I used as a functional unit, gene, refers to a specific substring within the DNA that encodes a very special kind of functional unit in biology known as protein. I'll talk about the exact encoding in a moment, but this is the basic definition of a gene. And obviously, when we have a string, there can be errors in the copying of that string. I'll just use the term mutation to refer to such a change in the sequence. There's all sorts of machinery that does things like copy editing to make sure that the mutation rate doesn't get out of control. But there's a small probability during the copying of any DNA string that a particular nucleotide will get replaced by another nucleotide in a given generation copying event. Questions? So one basic distinction that biologists make in talking about the underlying code versus the behaviors and properties of the organism that's produced from that code is we call all of this, the underlying code, the genotype. And we call the observed characteristics, how that organism behaves or looks as a result of the code that it's got, we refer to that as the phenotype. So, obvious point, there are some mutations that change the genotype, by definition, but which don't seem to have any effect on the phenotype. In other words, you could think of them as silent mutations, don't actually change any functionally observable characteristic of the organism. So that distinction has to be made between phenotype versus genotype. So I've already introduced the idea that there can be variation within this code, and this is referred to in general as population polymorphism. That is to say, when a change or mutation occurs within a population, initially, obviously, it occurs in a single individual, right? And maybe sometime later on, perhaps it proved to be beneficial, it was selected for over evolution, maybe it took over the entire population. And so now the entire population that used to have an A now has C at that position. We refer to that process as so-called fixation. So the two possible fates of an initial mutation are eventually it could become either lost, in other words, completely vanishes from the population, or fixed. That means it now is present in 100% of the population. And when it's intermediate, say, 50% of the population, we refer to this as polymorphism. That means there is variation from individual to individual within the population at that specific site of the mutation. There are many kinds of mutation. We will restrict ourselves typically to quite simple mutation cases. The simplest is a so-called single nucleotide polymorphism. That's a very long phrase for saying only one letter changed. Okay? So this is the simplest possible change in a DNA sequence, and it's actually the most common change within genetic populations. Lots and lots of single nucleotide polymorphism. An important characteristic of a polymorphism that I've already referred to is what fraction of the population actually has that mutated form as opposed to the original unmutated form. We refer to those two different forms in genetics as alleles. So there's a minor allele and a major allele, the minor allele being the one that's at lower frequency. And we refer to the allele frequency as the fraction of the population that has that specific change. Any questions? Like I said, it's pretty simple stuff, but we'll use it again and again, so good to know. When we go to organisms that have two copies of their genome, I think you can immediately see that we now have 4 possible cases for our possible pairings of our 2 chromosomes. In other words, you can think of each chromosome copy of that gene as having a 0 or a 1, depending on whether it doesn't have the mutation or it has the mutation, right? So biologists refer to the effect of a mutation under those possible cases as either recessive or dominant in their effect on the phenotype. The classic example is that if a mutation basically breaks something, in other words, it takes something that used to work and now it doesn't work quite right, you have another copy that works right. Usually having the one defective copy really has no effect on the overall function of the organism. Why? Because it still has one completely normal functioning copy. Okay? This is referred to as a recessive mutation. There are rare cases where a mutation present in just one copy will go and change the overall behavior of the organism. I won't go into detailed mechanisms, but this is referred to as a dominant mutation. The key distinction is a recessive mutation is basically silent when it's present as only one copy. It has to be present in both copies in order for its phenotypic effect to be observed. Whereas a dominant mutation only needs to be present in a single copy in order for its effect to be observed. This pattern is referred to as Mendelian inheritance. This is what Gregor Mendel figured out, basically the discovery of basic genetics that led to the development of the whole field of genetics. So how many of you know a human clone? Yeah, some of my best friends are clones. So, while it's like all terms in science, they get kind of taken over by the media and movies. Its meaning has been drastically changed to imply some kind of demonic power. But clone is actually a pretty trivial thing. It just means 2 individuals that have the exact same DNA, exact same, genetically indistinguishable. And this happens all the time. I mean, identical twins, that's what they are. I actually know a pair of twins who are both bioinformatics researchers. I don't know how, independently they both arrived at becoming bioinformatics researchers. Maybe there's some genetic predisposition for going into bioinformatics research. At any rate, in biology, this is quite important. For example, if you're working with bacterial cultures, the standard way of studying a bacterium is to dilute it down to very low density, plate it out on some kind of solid medium, food basically for bacterium, and if only one bacterium lands every square centimeter or so, it'll start growing, right? With no other bacteria around it. It'll grow and it'll grow and it'll grow, and eventually you'll see a visible colony of bacteria that is the location where that one bacterium landed on the plate. They'll all be clones, so they're all genetically identical of each other, and so you can study them, and you're studying one bacterium in terms of their genetics. So when we look at how the genetic material actually works in a cell, a critical distinction is between DNA and RNA. RNA is essentially an active form of the information that's stored in the DNA, basically just as kind of long-term storage. You actually want to use that information, as I'll describe in a moment. The first step is called transcription. It basically means that you copy the DNA sequence into an RNA sequence, more or less in exactly the same way that you would copy one strand of DNA into another strand of DNA. There are special molecular machines that do that work, so-called DNA polymerase, or so-called RNA polymerase. The key to this process, I already told you. That is to say that the individual letters base pair more or less automatically. They have a chemical affinity for each other. So what that means is these molecular machines basically just do that process very efficiently. Make sure that as you add nucleotides, which is the biologist's term for the individual letters, to the growing copy of a new chain, that you put the right nucleotide that complements the nucleotide on the parent strand. So DNA is, as I said, double-stranded. RNA typically is single-stranded. This has a structural form that is the picture you've always seen as the double helix. That is to say, it consists of two chains wound around each other, actually in opposite orientation. Okay, so the tail end of one is matched to the head of the other, and vice versa. Which means that given any sequence of one strand, you can automatically read off what the sequence of the other strand will be. In other words, you go to the end of one strand, let's say T in this case, that means the other chain will start with the complementary letter A. This relationship is referred to as reverse complementarity. Okay. So this is really, really useful. Why? Basically means that you can use DNA as a detector for detecting things, other specific pieces of DNA that you want to detect. All you have to do is take the DNA that you want to detect and maybe label it with something like radioactivity, fluorescence, and then just mix it with some other DNA. And if there's any piece in the other DNA that is matched to your piece of probe DNA, they'll bind to each other automatically by reverse complementarity. Okay? They'll find each other out of the whole mix, basically through chemical diffusion and then sequence-specific binding. Okay? So this is actually the basis of the entire biotechnology industry in a sense. The ability to find or decode individual pieces of DNA by making reverse complementarity do the work for you. A very simple example of that, so-called DNA microarrays. This is basically a glass chip with individual dots of probe DNA that are each synthesized to have an exact sequence of interest. Typically, something that's designed to match a gene. Okay? So by laying out a lot of spots on a tiny little array, you can have a probe for every single gene in the human genome. Actually, many, many different targets within the genome. Now, you take your DNA of interest that you want to measure. Well, how active was this particular gene in this particular sample? All you have to do is fluorescently label that sample, incubate it on top of the microarray, and the piece that matches that specific probe DNA will automatically come here and bind. Okay? So if there's a lot of it, you'll see a bright spot because a lot of it came here, and it's fluorescently labeled, so you'll see the fluorescence. If there wasn't any DNA that matched this particular probe location, you won't see any fluorescence because there wasn't any to bind there. Okay? The place where all this turns into biological function typically is the making of proteins. And it's a very simple encoding. As I mentioned at the beginning of the lecture, every 3 letters of the DNA encodes 1 letter of protein. This is a so-called codon of 3 letters in the DNA that selects one of 20 amino acids. There's only 20, so it's a 20-letter alphabet instead of a 4-letter alphabet. You can see why it had to be 3 letters of DNA. 4 to the 2 is only 16. 4 to the 3 is 64. Okay, so we use 3. And it's a universal code. All organisms, all of life, from the bacteria on up to us, use the exact same encoding of 3-letter triplets to amino acids with some very minor variants. The process of actually producing the proteins that go and do all the work in life is 1, copying the DNA to RNA, that's transcription, and then translating the codons in the RNA into amino acids in protein, that's called translation. Okay? So it's basically making a long string into a somewhat shorter string with a bigger alphabet.